So before I discuss transcription, we're going to go over some key terminology. So here we have the anti-sense strand, which is also known as the template strand, and here we have the sense strand, which is known as the coding strand. And these are the two strands of the DNA. The mRNA that is produced during transcription is complementary to the anti-sense strand, and the exact same as the sense strand, with the exception of uracil bases instead of thymine bases. There are three stages in transcription: initiation, elongation, and termination. Initiation begins when the RNA polymerase, the main enzyme in transcription, binds to a region on the DNA called the promoter region. There are two sets of promoter sequences shown here. They are both important. Binding to the promoter region ensures that the RNA polymerase binds to the correct strand on the DNA and in the correct orientation. When the RNA polymerase binds to the DNA molecule, it unwinds the part of the DNA helix to be transcribed. mRNA is synthesized in the 5' to 3' direction based on the template strand. Each new nucleotide is added to the 3' OH of the previous nucleotide. As I stated before, mRNA is synthesized in the 5' to 3' direction. As soon as the RNA polymerase starts to move along the DNA, another RNA polymerase can bind to the promoter region and start to synthesize another mRNA molecule. Therefore, many RNA molecules can be made at a time. RNA polymerase synthesizes mRNA much faster than DNA polymerase catalyzes DNA replication, mainly because RNA polymerase doesn't proofread. In termination, the RNA polymerase reaches a specific nucleotide sequence in a template DNA. It signals it to stop transcription. The RNA polymerase will detach from the DNA strand, and the mRNA strand will be released. Then the DNA double helix will reform. In prokaryotes, the mRNA is already ready, but in eukaryotes, the mRNA produced after termination is called the precursor mRNA, and this means that some modifications need to be made. The first of these modifications is the addition of the 7-methylguanosine cap at the 5' end. The next modification is the addition of a poly A tail at the 3' end. This is the covalent linkage of a series of adenine nucleotides. Both the 5' cap and the poly A tail allow the mRNA to exist longer in the cytoplasm. The next modification is splicing, which is similar to cutting and pasting. So here, the non-coding regions, which are the introns, are being cut, and the coding region sequences are pasted together. These are accomplished by the spliceosome complexes, which are made of snRNA, and SNRPs, which are proteins. So once the exons are spliced together, this forms the mature mRNA. In some cases, not all the exons are spliced together, and this is known as alternative splicing. This is how one gene can code for more than one protein.